everyone. Thank you for coming back in. We'll get started on our luncheon presentations. If you could come in, find your seats, that'd be lovely. And I'd like to introduce our first lunchtime speaker, Andrew Sato from Twist Bioscience. Andrew? Aaron, sorry.
All right, great. Well, those lights are bright. How's everybody doing? So today I'm going to give you, again, my name is Aaron Sato. I'm going to give you an overview of our new integrated offering at Twist Bioscience. And I'll talk about our new team, which is called Twist Biopharma Solutions. I'll throw in some case study data showing the power of our platform, but again, it'll be a whirlwind tour. So Twist Biopharma Solutions is the merger of Veris, which is, again, world-class in vivo antibody discovery platform using their hyperimmune mouse, as well as Twist Biopharma, which brought on board a lot of synthetic library capability. I'll also talk a lot about today how we use AI and machine learning to help us curate our data as well as find more leads, and also think about the future in which we can actually use AI and machine learning for antibody optimization. So our new branded group is now called Twist Biopharma Solutions. But as usual, I like to start with the one thing that Twist is really good at, which is our ability to print DNA on this amazing silicon chip. Kind of think of it like an inkjet printer, but rather than printing colors, we can actually print nucleotides. And on this chip, we can actually print up to 1 million individual oligos up to 300 base pairs in length. And then we use those oligopools to make all of the custom DNA products that Twist sells, from oligopools to cloned oligopools, to gene fragments, to custom clonal genes. But the real power I see in the platform is actually our ability to use oligopools for highly diverse synthetic libraries. And again, that was the foundation for Twist Biopharma as it became part of Twist Biopharma Solutions. And so, Twist Biopharma Solutions, please see us as your service arm of Twist to help you discover and optimize antibody drugs on your path to the clinic. And so really thinking about the merged organization of Twist Biopharma Solutions, again, it starts with high quality antigen that can feed into our system and services. And again, you can really pick and choose whether you want to use an in vivo approach using the hyperimmune mouse or other animal sources of immunization and single B cell cloning or hybridoma, or you could go down the more synthetic route using our fully human synthetic libraries and phage display. And as I mentioned, we're also doing a lot of work on AI and machine learning to help us find even more leads from our data, as well as help us with antibody optimization. Since we're a DNA company, we're also a powerhouse in the make test cycle. So we can make hundreds and thousands of DNA sequences encoding antibodies. And we also have a high throughput IgG service where we can also make hundreds and thousands of antibodies as well. So we really have the best of both worlds. We have amazing technology to help us discover and optimize antibodies, as well as the ability to make and test hundreds of thousands of antibodies in the back end. So let's start in and talk about our in vitro approach. Again, I've talked about our Twist Biopharma in vitro approach in the past, but I'll briefly go over it again. So again, we use our silicon platform to make oligopools encoding for specific, in this case, human CDR sequences. And we can make pools anywhere from, say, hundreds of oligos, even up to 1 million oligos in a pool. And then we can use those pools of CDR oligos, for example, to actually make fully human antibody libraries encoding for natural human sequence. And as an antibody engineer, I love using oligopools for generating fully human libraries because all of the antibodies that come out of our libraries are fully human. We only use fully human sequences in our CDR oligopools as we build our libraries. So even though we're making synthetic libraries, the repertoire and antibodies that come out of it are very naturally derived. And versus a lot of our counterparts that use synthetic libraries, either NNK-based or TRIM-based oligos, oftentimes can introduce non-natural amino acid distributions into their antibodies. So in terms of our overall offering on the discovery side, we now have over 20 antibody libraries in phage display derived from this in vitro library platform. We have both scFv, Fab, common light chain Fab, as well as VHH libraries. On the VHH side, we actually have over 6 different VHH libraries derived from either llama, camel, or humanized diversity. And then on the fully human antibody side, we have both, again, scFv and Fab-based libraries that you can use for developing fully human IgGs as well. In terms of our overall timeline, we can go from receiving your target to actually handing you back sequences as well as all the characterization data in basically 3 to 4 months time frame. So in terms of our overall process, I won't get into all the intricate details, but it's suffice to say we can pan and screen on any antigen you wish, whether that's peptides, proteins, cell lines that overexpress your antigen. We typically do about 4 or 5 rounds of panning using libraries pulled typically based on scaffold types. So again, VHH, scFv, and Fab. If it's a protein-based antigen, we often do a clonal-based ELISA to pull out of our hits. And we've also been doing a lot of NGS sequencing at each round of panning as well to give us additional repertoire data that we can then use to use machine learning to actually give us even more hits to make and test. Once we have all that sequence data, again, we can feed that into the awesome DNA engine of Twist to enable us to make hundreds and thousands of antibody sequences, make them quickly in our high throughput IgG team, and then we can use high throughput SPR and FACS analysis to give all of our characterization and binding data. So just one little vignette for the use of our synthetic libraries. In this case, we actually worked on a very challenging ion channel target that we did in collaboration with LeadX Pro that actually made a solubilized recombinant P2X7 ion channel for us. And then using our VHH libraries, we actually pan and screen against this ion channel and actually came up with dozens of VHHs that bind with very high affinity to this ion channel. And we're pursuing them further in terms of functional assays. But in terms of their use for LeadX Pro, they're actually going to be using it for crystallization of this antigen, whereas we're going to be pursuing more of a therapeutic approach. So on the in silico side, we've been using machine learning for many different uses within Twist. Several years ago, again, I thought it was very important for us to start incorporating this into our workflow. On the library design side, we've actually worked with several outside companies now to actually help us simulate fully human antibody sequences that go into the design of many of our libraries that are part of our offering. In addition, as I mentioned, we also use NGS analysis at our multiple rounds of panning. And in doing so, we can use both unsupervised as well as supervised machine learning to give us even more sequences that we can pull out of our repertoire analysis. I had always thought that we could actually get all of our hits by just doing primary screening. But what I'm learning is that there's a lot of antibody sequences that we miss, that we leave on the table, that are in our NGS data that often don't come around through our multiple rounds of high throughput screening. And so this gives us even more hits to make and test. Really what I see at the forefront of using machine learning is really in antibody optimization. And we're finding that we can use machine learning to help us with antibody humanization, which is actually really nice to use with our in vivo offering. But in addition, I'm really pushing forward to see if we can use machine learning to help us come up with additional variants of specific lead sequences without actually having to go through a library build approach. If we can empower this and show that this can be used, we can actually get to optimized candidates much, much faster because we can feed them directly into that make test engine. And I'll show some data today how we're using zero and single shot language models to help us do this. And then finally, we're using machine learning to help us come up with developability models. We've actually accumulated a lot of developability experimental data over the years, and we're actually using that to see if we can actually create models where we can predict developability based on primary amino acid sequence of antibodies. So just to show a little data on our use of machine learning, again, I've been really pushing forward this idea that someday you'll be able to go to Twist and just input your antibody sequence, and we'll actually be able to give you back variants online that you can then make and test with Twist. And so this is an example where we took an optimized humanized candidate that we had internally and then applied a zero-shot model, which basically means we can look at, use a machine learning model of proteins or antibodies, feed this sequence into it, and the model can tell you which amino acids in your protein potentially would be likely to be mutated to another residue within the natural repertoire. Again, this particular type of model doesn't require any additional data except for the initial input antibody sequence. And so from this initial model that we had, we fed this particular antibody sequence into it, and we were actually able to find that we were able to see pretty modest but significant increases in affinity relative to the parental sequence. But if we push this a step further and actually do a few shot model, where we actually not only give it the input antibody sequence, but also some additional variants that are similar based on some previous work we had done with this candidate, both negative and positive data, we can actually come up with even more impactful variant sequences derived from this initial humanized candidate. And so from this analysis, we actually showed we're actually able to increase the affinity up to by 45-fold relative to the parental humanized candidate. So again, we can do this very fast. We don't have to go through a library build, and we can come up with these sequences and directly feed them into our make test engine to give you back humanized candidates. And we're continuing to refine our models to make them even better, both for zero-shot as well as single-shot language models. So now I'll jump into the final part of our offering, which is our in vivo offering. Again, at Veris, before they merged with Twist Biopharma, were already experts in utilizing animals to discover antibodies. Their primary technology was using the hyperimmune diversimab mouse, which has an autoimmune state that can actually find antibodies to a wide collection of epitopes within a wild-type background, mouse background. They also can discover antibodies from alpacas and rabbits, and they're trying to push into other species as well. And they can also work with, of course, all of the other humanized antibodies out there, because we've been pre-qualified to use many of them in-house. We have a lot of expertise in doing a wide array of different immunizations, and this has also enabled us to tackle a lot of difficult to drug targets using this in vivo approach. They really have two paths that you can use. Of course, you can use the classical hybridoma approach, and I'll give you some timings for that in a second. But they're one of their other great strengths in the Twist Boston site here at Veris was really their ability to use the Beacon platform for single B-cell cloning. And as you know, the Beacon platform can actually be used to do multiple assays on each single B-cell. So you can actually accumulate a lot of data up front on each individual clone before you actually come up with the individual sequence for that antibody. In terms of the overall timing for the two different offerings within the in vivo offering, of course, you can rapidly discover antibodies using the Beacon platform. And once you have high titer in the animal of interest, within a 1 to 2 week time frame, you can very quickly get to sequence of those particular single B cells. But of course, you can always use the hybridoma approach as well. It takes a little longer, but again, it's lower cost and lower throughput, but you can still potentially get to reagent antibodies that might be useful for your particular application. Just a little bit of data on the power and expertise of this team. They've really shown that they can tackle really difficult to drug targets like GPCRs, shown on the left, using, in this case, DNA-based immunization. We've also shown that we can do cell-based immunization in the diversimab mouse as well, that's shown in the middle. And then finally, a really nice case study in terms of the ability of the diversimab mouse to actually find unique epitopes. They were actually able to, time and time again, actually immunize with an antigen that has 100% mouse-human homology and show they can get high titer against this antigen. Again, showing that the autoimmune state of the diversimab mouse allows them to find antibodies to particular epitopes that are oftentimes excluded in other mice. And then just getting again to the power of using the Beacon platform, you can use multiple serial assays on the platform. So you can look at both cell binding, ligand blocking if your particular antigen, your particular antibody, you want to block a specific receptor ligand interaction. Of course, you can do a whole series of multiplexed assays looking at, say, mouse, human, cyno cross-reactivity. And then they can even build in functional assays like internalization assays into their offering as well. So I find that the Beacon platform is very powerful for doing single B-cell cloning because it gives you a lot of data up front on every single B-cell clone in your immunization. And then this slide really brings the message home that time and time again, over the years, the team at Twist Boston has actually been very impactful in finding antibodies that have a wide range of affinities, and then definitely in that 10 nanomolar below in terms of monovalent binding. So really showing the success of this team to find hundreds and thousands of antibodies to our customer targets. So just bringing it home, again, we have these 3 technology platforms: in vivo, in vitro, and AI machine learning. They all feed into our ability to come up with hundreds and thousands of sequences potentially for your project if you combine multiple technologies at once. We have the power of Twist to make lots and lots of DNA as well as antibodies, which I'll show in a second. And then we have amazing instrumentation for doing tests on all of those antibodies. Those of you that, of course, work on antibodies, I'm sure, even myself, before I joined Twist, DNA and production of antibodies was always a bottleneck for me. So one thing I really wanted to do when I started the Biopharma team was really enable a new product within Twist, and that was our high-throughput IgG offering, because we realized that a lot of our customers actually use us for making antibody genes. So why not help them and actually make small amounts of antibody as well to help them with their screening efforts? So today, we can actually do 1 mil or 8 mil transient transfections for you in HEC-293, protein A purify them, and within, say, 20 days, deliver those back to you. We can also, again, do scale up to 8 mil to give you upwards of 1 milligram of antibody. And I use this service internally. Actually, my team in South San Francisco doesn't actually make their own antibodies. We can actually just give this as an order to the high throughput IgG team. So similar to us, you can also use this service to help you to make hundreds and thousands of antibodies for your screening efforts. And we're working really diligently to offer up CHO in the near future to not only make antibodies in HEC, but also potentially Chinese hamster ovary cells. And then once we have those antibodies, we have a lot of state-of-the-art instrumentation for doing characterization of all those antibodies. Of course, we have Cartera for doing high-throughput SPR. We have all sorts of high-throughput FACS analysis that we can do, as well as a whole team for doing really high-throughput as well as primary functional assays if your particular project requires it. As I mentioned, we also use AI machine learning to help with humanization. Since all the antibodies that come out of the Diversimab platform are wild-type mouse sequences, it's actually a really nice marrying of the 2 technologies where we can actually take those input mouse sequences or sequences from, say, alpacas, and actually very quickly humanize them using an AI machine learning platform that we've created at Twist. In addition, we also have our Tau platform, which again came from the Twist Biopharma offering, where we can humanize particular sequences as well as optimize them in a library-based approach. So if you either have a humanization campaign that you need to do or a humanization coupled with optimization, again, Twist can help you with that. And shown here is just a little bit of data where we actually took 3 different therapeutic antibodies. We went back to the mouse sequence that they were derived from, rehumanized them with our platform, and then compared the antibodies that we humanized back with the therapeutic candidate that was humanized. And we found that we were able to get very similar humanized candidates relative to those human therapeutics in the clinic in terms of their overall affinities relative to the chimeric antibody. So overall, in terms of all 3 offerings, hybridoma, single B-cell cloning, and phage display, on the single B-cell cloning and phage display workflows, they basically have very similar timeframes in their ability to get to sequences in the 1 to 2 month time frame, but eventually be able to get to a full package of characterization within a 3 to 4 month time frame. Of course, the hybridoma workflow takes a little longer, but as I mentioned, it has a lower price point. So overall, the combined Twist Biopharma Solutions team today has a full suite of leading platforms under one roof. We can take your initial antigen that you target you come to us with, and we can use all the technologies I talked about today, as well as all the optimization technology to discover your lead, as well as to optimize it for greater potency, as well as potentially humanize it. Once we have those candidates, we can do thorough analysis for characterizing your leads. And again, we can often go through multiple rounds of optimization if it requires it to give you development-ready drug candidates. So I'm really proud and pleased to actually introduce what we're calling our new gold standard within Twist Biopharma Solutions. The idea is that we really want to give our customers multiple shots on goal. And so ideally, kind of similar to how pharma approaches targets, when you come up with a specific target you want to go after, you want to hit it with multiple technologies. And because Twist is one of the few companies that has a lot of technologies under one roof, we potentially can do parallel paths of discovery against your particular antigen. And again, the idea is that each particular technology might have dominant epitopes on your target that might be a focal point for that technology. But by combining them together, say in vivo, in vitro, and in silico, we typically potentially can have, cover the antigen in terms of the potential epitopes that we can find against your target and really find the key functional epitope that you want to use for your particular therapeutic. And then the final thing that we want to add into this offering is a guarantee that we will use every single technology that we have to enable you to be successful in your project. And if we aren't, we'll go back and repeat many parts of the campaign to make sure that we actually are successful in the end. And this just gives you an idea of how we might do that in terms of the parallel tracks within the gold standard. Again, you might think about doing, say, a beacon campaign coupled with a synthetic library campaign. We can always use AI machine learning to help us with lead picking. We can, of course, do humanization of any mouse-derived antibodies that come out of the beacon campaign. And then finally, we can port that and make hundreds and thousands of antibodies as part of our Twist make test cycle. So again, by doing parallel tracks, we can ensure that our customers will be successful in the end. And again, we would, as I mentioned, apply a guarantee as part of the gold standard. So just as a little case study for what a gold standard might look like, actually, this is a nice kind of corollary with yesterday. I think Andrew actually gave a nice overview of kind of AI machine learning, the black box approach. And one question he actually had is, have people actually compared an AI-based, in silico-based library with other library-based approaches to see that it actually had some advantage over the other library-based approaches? And so actually, it just reminded me that I actually had some data that actually showed some of that. So what we did is we took a very difficult to drug target, C5a receptor, which is again a GPCR target. We did 3 different types of discovery workflows. One, we did an in vivo-based approach where we immunized the diversimab mouse with, in this case, DNA that encoded for C5a receptor, and then created both an immune library as well as a synthetic library as part of that immunization. We also used our in vitro libraries derived from the library of libraries as well to pan and screen against C5a receptor on cells. And then finally, we actually worked with another company, Macromoltech, to actually in silico discover antibodies to C5a receptor on multiple sites. And they actually gave us over 10,000 different putative binders for C5a receptor. And then we used those 10,000 putative binders to actually create a synthetic library where we shuffled that diversity. We took all of those libraries into a panning approach using both cell-based C5a receptor as well as protein-based antigen. And then we did primary cell binding as well as functional assays on all of the antibodies that came out of these campaigns. So from this analysis, we'll really be able to see which library-based approach was the most impactful against this particular target. So in terms of cell binding, we found high-affinity cell binders from all 3 approaches. As you'll see in a second, there was one clear winner of the three approaches that actually had a preponderance of the cell binders as well as functional antibodies. But in general, we're able to find high-affinity cell binders that bound to C5a overexpressing cells versus the HEK background. We also did, again, full FAX titration on all the hits, again, to get putative EC50s for all the antibodies. And we also did some SPR analysis, again, on the Cartera using C5a receptor micelles. Again, shown here on the right is just some example data where we, again, can get some putative KD values derived from the SPR that can give us a nice ranking of the different clones based on overall affinity. And again, that antigen was made for us by G-Clips. And then finally, this is just showing you one example of data, one of the functional assays, but we actually put them through a whole battery of different functional tests. But in beta-arrestin, we also showed that many of the antibodies actually had functional antagonism beta-arrestin in this particular assay. Again, indicating that these antibodies might be good to move forward in a therapeutic indication for a C5a receptor antagonist. So just to get to the summary of all the data, so in the end, all three approaches led to at least one functional antibody, but it really was the AI-generated modeling-based approach to generating a library that was actually the most successful in terms of giving us over 7 functional antibodies against this particular target. So again, putting it head-to-head, a naive library, an immune library, and then an AI-generated library. In this case, the AI library actually was the most impactful against C5a receptor. And again, this is an example where Twist would use multiple technologies in parallel to help and make sure that you were successful. In this case, let's say the customer was actually going after C5a receptor. We would have been very successful in delivering back to them a candidate. So just to conclude, Twist Biopharma Solutions really increases your shots on goal. We have huge breadth in our multiple technologies that we can use. Of course, we have huge expertise and quality that we can bring to the table to help enable you to be successful in your antibody discovery and optimization campaign. And then finally, we're very fast. We have this amazing ability to rapidly discover and optimize antibodies, but at the same time, we can rapidly use our DNA platform to help you make and test lots of different candidates for your particular project. So just to conclude, Twist over the years, combined with now Averis, has hundreds of successful antibody campaigns under our belt. We have hundreds of partnerships across multiple therapeutic areas. Twist Biopharma actually now also has multiple candidates that have been derived from our platform in the clinic, and we really have the marrying of 3 awesome platforms to now deliver to you, and we would love to do that within the context of our new gold standard offering. And I'd be happy to answer any questions.
Hi, Erin. Thanks very much for a really nice update. I was wondering, with regard to the AI ML approach that you take, do you also look at the diversity of epitopes that you get from that as well?
So the library-based approach I talked about actually did take into account multiple epitopes. And so they actually targeted, I think, up to 10 different epitopes on the target, and they tried to predict specific antibodies that would bind each of those different epitopes.
Thanks.
Nice talk. I was curious about the first part of your talk on the 0-shot and few-shot examples you gave us. You got affinity 2-fold to 45-fold. I was wondering, in terms of same question, I guess, does it change the specificity and the epitope coverage of your affinity matured variants?
No, again, similar to any antibody optimization campaign, we would make sure that there's no epitope drift. So in these cases, there was no epitope drift, but that's a great question. We don't want to apply additional mutations that actually cause it to drift to another epitope, and in this case, they didn't.
Quick question about that last, the structure-based neural network, where you discovered about 15 hits. How big is the library, or is it like a combinatorial library, or is it a...
Yeah, great question. I mean, all of our libraries are on the order of 10 to the 9th to 10 to the 10th, based on kind of the limitations of phage display. And again, that's a typical library size for both our discovery libraries as well as our optimization libraries.
Got it. So then are you basically using the model to give you the distribution you should screen in the phage display?
We're using the AI model to really give us the initial starting point for our library design. So from that collaboration, they gave us 10,000 antibody sequences that were putative binders to C5a receptor, and then we used those 10,000 sequences to design the library that was then at the 10 to the 10th level.
Thank you.
Yeah, so the high throughput AGG is very interesting, especially when you apply language models to try to learn which variants will work better. But I'm wondering, there are these models like Rosetta Diffusion, AlphaFold, where you can do structure-based approach to predict specifically which variants would work better based on the structure. And I was wondering, to hear some of your input about how do you see these models? Because in theory, they should be much more powerful than language models because they have the inductive bias about the 3D structure.
We haven't really gotten yet to docking of antibodies with antigens and see how the variants from the language model are actually impacting those interactions. That's a great great idea. We are working to see if we can rapidly model both antigens as well as antibodies. And we plan to get into modeling next. And so we'll definitely do that, but we haven't gotten to that yet.
Thank you, Aaron.
Great. Thank you so much.